in front of a live studio audience. Hi guys, thank you once again for tuning in. Last week I kind of left you guys hanging with the tissue culture stuff. We're going to do a continuation of that, as you may be aware by what I have sprawled across the lab table. But first I just wanted to mention a couple of quick things. One, my phone video camera will shut off after the video is 35 minutes in length. That's why there was this cut off the last video. No biggie, just figured I'd mention that. A couple of quick orchid updates. One of my orchids, one of my Phalaenopsis, died of some kind of weird infection. The leaf kind of lost its color and then oozed a weird brown fluid, so I just threw it out. My dehydrated orchid is recovering. I wrapped its roots in wet sphagnum and put it in a bag. Generally, I would advise against restricting the airflow to an epiphyte's roots, but because of how bad a shape this plant is in, I'm just allowing it to recover. It seems to be doing okay right now, but I'll keep a close eye on it. I have also been repotting several of my epiphytic and two of my terrestrial orchids just because they've outgrown their pots and it was time to freshen up the media. That's about it as far as the plants go. So I started last week with what tissue culture is, why we do it, and how you can kind of teach yourself how to tissue culture. I'm about to get ready to sow some Cypripedium and Goodyear seeds. I may make a separate video of me actually doing that process, but before I get to any of this, I would like to kind of go over some of the orchid-specific tissue culture stuff. So I think last week I had shown you guys all of the different media components. You have agar, and you have charcoal, you have various mineral salts and sugars and hormones and a variety of other things to induce orchid seeds to grow. I have all of this pre-made media here. Again, I don't like to pre-make it very much in advance, maybe a few weeks, but generally not more than a few months. I am going to be sowing several Cypripedium species, and you can see they differ in the amount of potato and amino acid material. So this would be for pubescens, this would be for Californicum, there are some for Reginae, and then I have a variety of other medias. They're all labeled because I don't know if you can see this, but it just looks like a black gel, and it's not like these are totally different media. So this is P669 or P668 added banana powder, and this is Knudsen 400 media. Totally different medias, and basically I was talking about how Phytotech has a whole series of products for all of your orchid needs. I'm not trying to promote anybody, but it's where I get a lot of my stuff from. There are a variety of medias for various plants. There are different ones for epiphytes. There are different ones for terrestrials. Some of them are more general purpose. Some of them are specialized for particular groups. You can have orchid alliances where there are several genera that are closely enough in relation to each other that they can be kind of culturally classified as the same. And they have almost identical seed growth requirements, and they can be crossed between one another to form intergeneric hybrids. That's where you get the BRRA, BLLA, and all those other guys. But that is something to keep in mind. I wanted to quickly talk about green pod versus mature seed preparation. So I made a nice little flow chart up there, and I'll get to that in a sec. But there are a couple of things about tissue culture. First, the sterilization. I'm going to quickly show you my glove box here. I have a whole bunch of things. All of this stuff has been through the pressure cooker, except for this. This is pod bleach. I didn't label it, but I know what it is. I have a thing of ethanol here. I have a cover so it doesn't evaporate. I have a pipette full of the ethanol in there so it stays sterile. I have some of my tools here. I'll have to grab some tweezers before I get going with this. I have this glove box where it's completely sealed until you open it. I have a bottle of ethanol in there. There is a thick, I'd say that's about an inch deep layer of 20% bleach. And then there's a platform for me to actually do my work. So basically all of this stuff that I'm going to be putting in here has been sterilized on the inside. And when this goes into here, I'll put it in there. And then once everything is in, I will use the ethanol to spray the inside of this with it all shut so that it all becomes sterile in there. And I'll usually roll these around in the bleach so that everything is surface sterilized and is ready to go. And again, I'll let you guys see me do that in action at some other point. But that's the gist of how this thing works. What I do after I put everything in is I put the aluminum foil around the seams here after spraying it with ethanol so that no air can get into here. And I'll also clamp it with these. I have 4 of these. Whoops. These are just paper towels that I've pressure cooked. And the reason I do that is because I want to be very thorough. And this seems to work fairly well. The other important thing, I usually store this thing over here. And I cover it with this blanket so that light doesn't hit it, because UV light will degrade alcohol and bleach. The type of alcohol really influences whether or not light degrades. I don't know what the effective light on ethanol is, but you can never really be too careful. So I try to avoid the UV light by covering everything with this when I'm done. And then we have just all of our tools. So there is something called green potting, and green potting is where you take a nearly ripe seed pod from an orchid. And, oh, by the way, for the person who I sent a couple of weeks ago's Orchid Chronicles video to, who said I need to have a tripod to stabilize my hand when I'm holding the camera, this is for you. Okay, anyway, so everything is labeled, whatever. The orchid seed pod, before it dehisces or splits open, is sterile on the inside, and you need it to stay sterile. What people will do is they'll take a seed pod that is due to ripen within the coming, month, 2 months, depending on the species, and they will remove it from the plant, surface sterilize that, so they can just cut it open and put the seeds on the media. And this is called green pod method. Now, there are some pros and cons to this method. The pro is that you don't have to worry about treating the seeds. They're already sterile. And in some instances, you might get these seeds to germinate much more quickly. There are a few disadvantages, however. One major disadvantage would be if it is a plant where the seeds have germination inhibitors or dormancy. It's better to use mature seeds because bleaching the seed itself will remove any germination inhibitors. That way you just end up with seeds that'll grow for you. And the other disadvantage is you may not get very even or high germination rates because you've interrupted the maturation process of a seed pod, although with a lot of tropical orchids, they generally will have been worked with enough so that people have an idea of basically how long to wait before you remove the seed pod. So let me just quickly show you my flow chart over here. When you put your seeds on the tissue culture media, do you want to give it light or do you want to keep it in the dark? Well, is it an epiphyte or a lithophyte? Lithophytes grow on rocks, but they're functionally epiphytic. If so, you want to give it light. Terrestrial orchids should not be given light in the seed stage. They have a requirement to be underground, although I don't know how far underground a tiny orchid you can get, but you generally don't want to give a terrestrial species light. It can disrupt or even prevent germination. There are some terrestrial species that will germinate regardless, but since we don't really have a grasp of which species, we don't know every species that falls in the category of it can germinate in the light, it just doesn't make a difference, and if you put it in light, it will die. So just to be safe, keep every terrestrial in the dark until you're sure that they're producing photosynthetic material. Is the orchid species temperate? If it's not, then you can green pod it. Again, not temperate, green pod. Temperate, you should not green pod it. You should allow the pod to dehiss, remove the seeds, and clean them. So, if you do that, what I've done here with a bunch of cypripedium seeds of various species, we have pubescens, Reginae, Californicum, and then Goodyear pubescens, which is obviously not a cypripedium, is I've taken some water and a material called Tween 20. It's a surfactant. It basically works like a detergent. A little bit of Dawn dish soap also works just as well for this purpose. And it basically helps the orchid seed become attracted to the water, if you will. You want to force the air out of the seeds. So what I did, very excessively as you can tell, well, I tried the regular centrifuge, but this kept popping open. So I moved everything to microcentrifuge tubes. I microcentrifuged all of the orchid seeds until they sank in the water with Tween, and then I just dumped out the supernatant. Some people use vacuum pumps for the same purpose. I just didn't have a vacuum pump. I'm sure if you're at home, a vacuum pump is more accessible. So apologies on that note. So I'm going to do a couple of things today with my orchid seeds. I don't know if I'm going to film all of it, but basically I have a couple of different medias that I want to try Goodyear on. And actually, let me grab the other one. I want to put some on. My bad. Let's see. It's gotta be in here somewhere. Just give me a minute. Man, I did not know how heavy that was gonna be. Jeez Louise. Right, anyway. I'm gonna need some of these in a little bit. Anyway, where is the, I already made that media. Is this, nope, that's not what I'm looking for. Why does that look contaminated? I don't think it is, even though it looks it. That is so weird. Okay, whatever. I thought that I had half strength. 668. Yep, got it. Okay. I need that. Actually, I'm going to move this away from the edge of the counter. Animals. Okay. So what I'm going to do is, after I bleach the Goodyear seeds, and by the way, surface sterilization takes about 10 to 15 minutes. The concentration of bleach, they say, I've read that 15 to 20% bleach is usually sufficient. The thing is, bleach varies in how much sodium hypochlorite there is, and lately people have just said, all right, 0.5% sodium hypochlorite is usually sufficient. For the bleach I'm using, because the bleach I'm using is about 8% sodium hypochlorite, I need between 6 and 7% bleach to make 0.5% sodium hypochlorite. And I have various concentrations of sodium hypochlorite here, because what I'm going to do is, first I'm going to use the normal 0.5% sodium hypochlorite with the Goodyear seeds and try them on these 4 media. Then I'm going to do the Reginae and Californicum in the 0.5%. And then I'm going to do the pubescens at these 3 different concentrations of bleach, well, of sodium hypochlorite, for different lengths of time. I was told by Bill Steele, who's a really cool dude with Spangle Creek Labs, who grows a lot of native cypripediums, you want to soak the Reginae for 35 minutes, Californicum for 45, and then pubescens, it varies. It's anywhere between 20 minutes and an hour, so I'm going to have some that are soaking for 30 minutes, some for 45, and some for an hour at the 3 different concentrations to see what gives me the best results. But, see, I have these little tubes of the bleach solution. So this is my 1% sodium hypochlorite. This will be for the pubescens. And one I will allow to soak for 30 minutes, one for 45, one for an hour. And you get the point. So this is, all of the stuff here has the Tween added to it so that it makes it easier for the seed to sink into it. The sterile water that I have here that I pressure cooked to rinse doesn't. So basically what I would do is I would get the seeds into the bleach, and at that point I would not allow outside material to get into the container with the bleach. Then I would get everything into the glove box, allow the glove box to be closed, sterilized, what have you, and then I would go ahead and get all the seeds on the media. What I generally do is if I'm using the loose seed, I will add enough extra of the sterile water here to float the seed over the surface and shake it or swirl it a bit so that the seed distributes. Then I'll use a sterile cotton ball to soak up the extra water. You'll lose a few seeds by doing that when you put the cotton ball down, but you have so many seeds to begin with, it's not that big of a deal. And that's how I go about doing that. And if it's a green pot, I'll usually just use something like this to kind of distribute the material along the surface of the media. I don't think I'm going to use a scalpel today, so I'm just going to move that out of the way. And the other thing is I try not to crowd the glove box. I don't put more in than I really need. It's probably better if I put one thing in at a time and then open it back up and re-sterilize. So before I end this video, I'm going to quickly take a look at the tissue culture stuff I already have going. All my epiphytes are in here. I have a cabinet over there with the terrestrials. So I have a Phalaenopsis, that's seed pod, dehisced pretty prematurely on me. No, sorry, it didn't dehisce. It the flower stalk was withering away, so I removed the seed pod and opened it, green potted it onto this. You can kind of just see some of the material down there. You can see what are probably the beginnings of protocorms. I have 2 vessels of that. I have one of my Paphiopedilum groups under the light just to see what happened. The other 2 flasks of them are not in the light. And then I have some Phalaenopsis from a seed pod that dehisced on me. Well, not literally on me, but I was hoping to green pot it, but it had other plans. And you can see the development of the protocorms on the surface of this media. They're getting to a state where they're relatively round. It's been about a month since I added these. I don't see any contamination. It would have contaminated by now. Those are all of the very small protocorms. They're just starting to fill out and become really round. Eventually they'll become green and photosynthetic. It's just a waiting game. They can take a while. So that's the gist of how you go about doing orchid tissue culture stuff.